The title of this talk is Place Cells Come to Brooklyn. In 1975, Jim moved from Michigan to Brooklyn, and I'll roughly be summarizing some of the work that was done there. Some of you may know Brooklyn, but this is what it looks like on a Google map. It's this darker area with everything else fading out. And the pin here is in the centroid, the weighted center of Brooklyn, and it's going right through Downstate Medical Center. And in the right, we see Downstate Medical Center, and there's the pin coming through, and it's going right through Jim's lab on the 5th floor of the physiology building. So he moved in, and I just added this slide, thinking that we'd misnamed this symposium, and I think it should have been named something like this. This is going to make John O'Keefe very happy, or so happy. And there are a lot of Jim phrases that are, they sit in your mind and you say, I think it means something. I'm not really sure what it means, but it means something. But one of them that stuck with me for as many years as I can, since the early 80s is Jim would come in, I'd be recording something, a nice place cell, and he'd get very excited. Maybe he first would say, this is going to make John O'Keefe so happy. But after that, he would say, this is cognition. And I'd say, that sounds deep. I'm not really sure what it means. And so I've been trying to figure out what it means for the last 30 or so years. But I think it is cognition, whatever that means. So Jim, moving to Brooklyn in 1975, started the Place Cell Lab, and it became the Place Cell Group. Not all were students, but they're colleagues and coworkers. And these are, I put this together, I didn't get everyone's picture, but these are a lot of the people who worked and did substantial work in the Place Cell Group. I'm trying to go a little bit quickly. This is a little goofy, but Jim, as we're seeing, Jim has a unique style. And I came as a postdoc with no background in this area, but immediately I was not treated as a student, but as a collaborator. He said, what do you think about this? And I would have no clue, but it was always a pure collegial relationship. It was peer-to-peer even when it really wasn't peer-to-peer. I never felt threatened or bossed or anything like that. Always a colleague and a friend. Another phrase I think that's very important that comes up frequently is the phrase, watch the behavior. And in fact, a few of us co-authored an interesting paper in the mid-'80s called Watch the Behavior, and it summarizes a neuroethological approach, which John O'Keefe talks about, which is, if we don't know what's going on, let's let the animal tell us what's going on, record from the cell, and watch the behavior, and look for the correlates, and the cell and the animal will tell you something you may not have been looking for. And I think that's what John described earlier, but it was also the key word, the approach that Jim used. Insightful. I'll skip stories and phrases. Well, anyone who's been around Jim knows about stories and phrases like, this is cognition. But the stories are not so short. But once the story starts, you may have heard it 10 times, but it has to run to its conclusion. And we can almost all recite some of these stories word for word. Now, contributions, we've been hearing about contributions, but let me try to put the contributions in context. And I think the general notion Jim had, well, he had extremely broad, from behavior and psychology to biophysics. And I think the motivating feature of Jim's work is to put place cells, or put these cells in the brain, and to relate these behavioral correlate levels things to identifiable cells, to the state of the network, to connections to other cells, and almost all of the work that he has contributed to involves those sorts of questions. So John earlier talked about Jim going outside of the hippocampus to look at the inputs, emphasizing we have to know what the inputs are to know what this is doing, and that's many of the steps he took. I remember Jim very, very clearly saying to Bob Muller and me, I'm moving to retrohippocampal areas. And he would begin studying areas outside of the hippocampus, looking for what's going on in the afferents to the hippocampus. And the theme of this, overall theme of this meeting is about head direction. And I think many of us feel that, if not the most important, the most notable thing we're aware of that Jim contributed was the discovery of head direction cells. I'm going to skip this quickly. This is about theta and complex spike cells, and John O'Keefe has introduced them already. These are from the early papers. Complex spike cells showing these decremental bursts that occur rapidly, frequently when the animal is walking through a firing field. And theta cells firing rhythmically with theta. But Jim always emphasized it. It wasn't really the rhythmic firing in theta that was critical. It was the doubling of firing rate of these cells, which he was guessing were inhibitory interneurons. And so thinking about the network properties of the system. Well, during theta, inhibition's increasing. What is that telling us about the whole network state of the system? Asking questions of that sort, separate from what's the specific signal going on here? What's this cell telling us about the brain? This is from a number of early papers. This is from Dr. Steve Fox's work, or first study with Jim Rock. But this was trying to make a mapping between theta cells and complex spike cells, which were describable from extracellular recording, and cell types in the hippocampus. So what they did, illustrated here. I don't have a pointer that works. I don't. Yeah, an electrode. This is a single electrode track from the study, and recording the different cell types. The T's are the theta cells they found at different depths. The C's are complex spike cells. And what they argued, and I think pretty convincingly, is that the complex spike cells were found in the pyramidal cell layer. So it was a pretty good piece of evidence that the complex spike cells, which were the play cells, were the pyramidal cells of the hippocampus, while the theta cells were not concentrated there. They were likely the interneurons of various sorts. As someone has mentioned, I came studying play cells. We initially did the paper that Phil was talking about, John was talking about, very videotape type recordings, but early on, working with Bob Muller, we collectively decided we were going to quantify these analysis. And in 1987, published what took several years to develop, a computer-based presentations of play cells. So Jim was the three of us worked together. I don't want to say one more than the other, but I think an early study showing that, hoping to convince people that play cells were not some figment of O'Keeffe's or Ronk's imagination, but they really existed. And from this earliest paper, we showed figures such as this, where play cells characterizing the firing fields. They could be in the center, they could be in the edge, they didn't seem to have any topographic organization within an environment. And importantly, some cells that are good play cells could be dead off in an environment. So why are they play cells? Because when we took this cell in the rat to a different environment, say a rectangular enclosure, it would be a pretty play cell. So the notion that a subset of potential play cells were the play cells in environment X and a different subset in environment Y. This emerged from these early studies. I want to be fairly quick, but the thing we are focusing on in this meeting is the discovery of head direction cells. And the first paper, as someone noted earlier, is an abstract in 1984, where Jim had really discovered head direction cells. And he rapidly put this to publication. So a mere 6 years later, it was in publication. And this is from that first publication, maybe the figure someone showed earlier, I don't remember. So why did it take 6 years? Well, in this case, developing and modifying the tracking system and sort of insisting, one of the three who I won't mention, insisted that we had to have the quantified procedure in place before publishing it. And at this stage, Jim was involved with drawing rats fairly well. I think the accuracy of drawing maybe correlates with when he was studying behavior. Pretty good rat. And what you see is on the rat's head is a red lead and rigidly fixed to a green lead. This let the camera overhead track two locations. And by tracking two locations with every sample, you get the animal's head orientation. And this permitted the tracking of head direction in addition to position. And this procedure really is largely unchanged in current head directional cell studies. And maybe I'm wrong, but I think it's what people are pretty much doing. It set the stage. And I want to talk about, so this 1990 paper, which I was embarrassed not to have read for many years, but reread this last week, this pair of 1990 papers with Jeff Taube and Bob Muller, and written by Taube, Muller, and Ronk. I was sort of amazed how much they covered besides just saying, well, there's a plot with head direction. They did so many cool, simple mini experiments which set the stage and end up being correct. So one of them is illustrated here, I actually had to take a frame out of a video to find a good overhead movie. And so the plots to the lower left are head direction by plots taken in 12 different regions of the apparatus. So here is roughly what the 12 different regions were that produced those head direction firing plots. And in all of those regions, the directionality was the same. So this was, I think, a pretty convincing sense that direction really is what's going on here. It's not angle to the cue card or something like that. It's really this broader sense of direction. Because the cue card was an important stimulus. In fact, if you rotated the cue card, they all rotated, but they didn't all point to, say, the left edge of the cue. They didn't always point to, say, the left edge of the cue card. It anchored the frame, but wasn't the driving stimulus. And perhaps the most remarkable finding was the occasions when they were able to record pairs of cells, which in the days of a handful of electrodes, before tetrodes, was a big achievement with hardwired window discriminators. Recording pairs of cells, head direction cells, the relative directionality of the pairs of cells was locked. It was locked here, in an example from the early paper, in the cylinder, in the upper frame, where the difference in direction, is that 100 degrees? I can't really read it, but it is fixed. And then taking the animal to a rectangular enclosure, the same relative position was locked in. And this was continued to be the case wherever you recorded these two cells, whatever the animal's doing, whatever environment the animal is in. So this represented a very, very different type of representation from the place cells, which remap with every environment, the way using current terminology, where for the head direction cells, there's one map. It's a circular representation, and I think this is sufficient data to argue for a one-dimensional attractor. It's enough to get us moving, and there's a lot of work that's followed this along, but nothing more substantial than seeing this locking of more than one cell to the animal's head orientation. And I think that summarizes what I wanted to say, and move on to who's next on our list.